of Yash Institute of Pharmacy, Chhatrapati Sambhajinagar, Dr. Dr. B. Gita ma'am completed her D Pharm and B Pharm from Madurai College with first class from the TN Dr. MGR Medical University, Chennai. B. Gita madam pursued her M.Pharm in pharmaceutical chemistry with first class and PhD in March 2012 from JSS College of Pharmacy, UT. Under the TN Dr. MGR Medical University, Chennai, she was GATE 2001 scholar with a 79.23 percentile. In starting phase, she worked as a hospital pharmacist at a Joseph Hospital, Dindigul in 1991. Then she worked as a tutor in Department of Pharmaceutical Analysis College of Pharmacy, Sri Ramakrishnan Institute of Paramedical Sciences, Computer, in the period 1997 to 2001. Professionally, B. Geeta Madam was worked as an assistant professor in the Department of Pharmaceutical Chemistry at RVS College of Pharmaceutical Sciences, Sulur, Computer, and Arkaya and B.M. Reddy College of Pharmacy, Bangalore, in the period of 2003 to 2013. She was recognized as a full-time research scholar under AICTE-sponsored QIP JSS College of Pharmacy, UT. She worked as a professor, head, and vice principal at a Chairman's College of Pharmacy, Coimbatore, and Swami Vivekanand College of Pharmacy, Illampallam, Tiruchingod, and Sri Ambirami College of Pharmacy, Coimbatore, since from 2014 to July 2023. Currently, B. Geeta Madam is working as a professor at SNS College of Pharmacy, Coimbatore, since 2023 to till date. Academically, she is expert in various subjects like pharmaceutical organic chemistry, medicinal and synthetic chemistry, and pharmaceutical analysis at a UG level. She has a good command on subjects like chemistry of natural products and modern pharmaceutical analytical techniques at a PG level. Her research interest area is the isolation of phytoconstituents using chromatographic techniques like Coulomb chromatography, preparative TLC, etc., and characterization of isolated constituents using spectroscopic techniques like UV, IR, NMR, and mass spectroscopy. She developed in vitro and in vivo methods to determine the potency of a plant extracts. Madam has a very good practical mind to develop and validate analytical methods for the assay of drug products by HPLC, UV, and colorimetry. She published more than 10 national and international papers on microwave assisted rapid extraction, pharmacognostical, phytochemical, and phytochemical evolution in vitro anti-cancer studies, analgesic, antioxidant, and hepatoprotective activities. She supervised 10 B Pharm and 8 M Pharm students. On her credit, she got Best Paper Award in Indian Journal of Pharmaceutical and Educational Research for the paper entitled Microwave Assistant Fast Extraction of Mucilages and Pectins. Also, she received Young Research Active Member Microwave Assistant Fast Extraction of Mucilage and Pectins. Also, she received Young Research Active Member in AVRS and PAA MMC MRB Faculty Award for the free MRB classes conducted online during the students' preparation organized by Pharmacy Alumni Association Madurai. Madam has participated in many state, national, and international level short-term training programs, seminars, conferences, and workshops. She delivered more than 10 guest lectures at national and international workshops. Uh, she delivered more than 10 guest lectures at national and international conferences. She was one of our Galaxy members who delivered online guest lecture in the national level webinar series on Trends in Pharmaceutical Sciences and recognized by Yash Institute of Aurangabad on the topic Significance of Phytochemicals Research in Drug Discovery on February 25, 2022. She has a professional membership of IPA, APTI, and registered pharmacist of the Tamil Nadu State Pharmacy Council. Thank you, ma'am.
एक सेकंड मेरे पास उधर रखते
for your wonderful introduction, and it increases my responsibility too. I'm proud to share.
Thank you, Amruta. All participants are requested to be interactive during this session. Your attendance will be monitored by the Cisco WebEx. If anyone have any technical glitch, type in the chat box. After completion of each session, feedback link will be displayed in chat box. So, without further any delay, me take this opportunity to welcome all the participants once again across the nation of this vibrant, energetic and motivational session ahead with Dr. B. Geeta Madam. Thank you, ma'am. Over to you, ma'am.
Ability more. And also, I would like to thank YAS Institute of Pharmacy for giving me this opportunity. And I thank personally my friend Gajanan, who has introduced myself to the principal, sir. So I thank the principal and IQAC coordinator members on behalf of SNS College of Pharmacy and Health Sciences, Coimbatore. By submitting my pronouns to Lordesh Saraswati, I'm starting my session. So I'll share my screen.
By two different devices, and your voice is giving echo. So will you mute or reduce volume of your another system?
Yeah, I'm not able to hear you. Could you please repeat it? Am I audible?
It is okay now.
Okay. So coming to the today's topic, that is natural products and drug discovery. So we are working with like we have all the products, especially whatever we are dealing with right from our day-to-day life. We are having morning till night. We are giving across the natural products. So whatever we are seeing in the natural, it is easy to accept, and also it is easy to digest and also get eliminated. So as a drug, when we call as a drug, it is a chemical moiety, either in the form of the synthetic or in form of the natural product. So when we take a natural product, we have different categories like plant products, microorganisms, fungi, bacteria, etc. And nowadays we are synthesizing from soil, if we are remembering about
इथे arrow नाही का?
so if we are remembering about the natural products, they have been born, they are having a traditional source molecules and modern drug discovery programs. You can see an extensive researches in the past. So earlier in the 19th century, if you take the drug marketed of natural products. So the natural product chemistry, why it has to be considered as a source in the drug discovery is it has a huge diversity of secondary metabolites. They vary from plants, microorganisms, and animals. So when we remember the drug, morphine and its analogs. So the discovery of morphine in 1815 and 1819. Why we have to consider these opioid analgesics have proven to be a best analyte in analgesic in case of accidental injuries, in case of surgeries. And when we take the structural elucidation, the chemical structures were not elucidated for many years. After the chemical structures were elucidated and they have been marketed as drugs, we could able to overcome their side effects by synthesizing semi-synthetic analogs. So there are some few examples quoted in this introduction part, like morphine, strychnine, and various other colored pigments which are isolated from the flower, like anthocyanins, they have been commercialized as products. So when we take in the 20th century, the OTC drugs were found to be from natural products, and when the chemical techniques and the chemical knowledge is becoming refined and sophisticated, we are able to synthesize many of the drugs from them as semi-synthetic moieties. So we can take more advantages from the natural products from. So coming to the next part, when we see about the microorganisms, discovery of penicillins from Alexander Fleming, who has delivered, after the discovery of penicillins, various infections, bacterial infections had been cured. And the penicillins, the beta-lactam antibiotics lay a foundation for other antibiotics such as cephalosporins and macrolide antibiotics. So when we remember the natural products, the drug either it can be isolated from various sources like microorganisms, plant products, and marine organisms, etc. So when we in this presentation, I want to highlight the importance of natural products, and as a researcher, we need to be more specific in each and every steps of the research. So I'm going to mainly talk about the steps involved in the phytochemical screening. So where we are, when this phytochemical screening, if it is still taken care, we would have come out with many other branches of research. Like I will come to you in my next slides. So these are some of the interesting examples. See how this aspirin, how we got the beautiful drug aspirin. So when the ancient Greek, when they were chewing this salix, they found that it reduces the fever and the inflammation. When they were able to found that there was a reduction in the body temperature and also pain, then this beautiful drug from the natural product has been converted into aspirin by the German company Bayer, and it has been marketed in 1897. So this is this part. This is the phytochemical part, which is an important source of natural products. So why we have to concentrate more on plants means, if you want to tell colloquially, I have heard about if you take any diseases, the adjuvants and the nutraceuticals are playing a better part in the therapy. So when you take, for example, you're having so much of tiredness, the caffeinated derivatives or drinks will make you more active. And in case you are having, we would have experienced in case of during our pandemic times, like how these plants and plant materials which are grown near our places, near our home, it serves as a home remedy. Every morning, every mother is a natural product chemist who provides or who serves the whole family with all the derivatives, constituents, phytoconstituent in a form of extract, either in the form of vegetable soups or in the form of an extract during cold and viral infections. So plants, why we need to concentrate more on plants? We are, as India is known for a well-known resource for natural products and plants, we have a tremendous source and we have like the statistics says only 10% of the whole plants has been evaluated so far. So out of which we are having valuable compounds, like they provide valuable compounds in case for the development of the new drugs. They act as a treasure for many of the new phytoconstituents. So if you are concentrating on the flavone and flavonoid constituents from the secondary metabolites, we are coming with beautiful moieties which are in the pipeline in the drug discovery. Why we need to concentrate more in the phytochemicals? Because the molecular architecture which is produced by the nature is cannot be compared with the synthetic molecules. So when you see the big structure like erythromycin, or when you see the structure like streptomycin, it is very difficult to get the lead as such in the synthetic laboratory. So they act as a molecular architecture which is accepted by the receptors. So in this way, they serve as a good lead in the drug discovery. And if you take modern techniques of isolation and elucidation using hyphenated techniques, we will be able to discover many products. And also the nutraceuticals and dietary supplements, they are going to take a big part in the next few years, because these nutraceuticals will prevent us, or else they will act as a prophylactic source from preventing us from many ailments. So as a whole, this slide covers the whole topic, like why we need to concentrate. As the whole world is back towards all the older things, golden things, the plants, they have invaluable compounds. They act as the treasure for the phytoconstituent. They have the molecular architecture which is unimaginary by the human brains, and we are able to get a molecule. So if we are able to remember like the structure of streptomycin or the macrolides antibiotics, we will be agreeing with this point. So if you take the beautiful drugs from the natural products, we cannot leave these point due to some tediousness in the research. So again, they have higher molecular weights and they have various chiral centers, which is proved to be biologically active, and their structural diversity makes us useful in various various treatments. And the secondary metabolites, they have the property of more friendliness than the synthetic molecules. Hence, this research in natural products has to be taken with more care in bringing out new moieties for the human health. Coming to the phytochemicals, they are a group of bioactive compounds present in all the part of the plant. If you take numerous phytochemicals, they are used to treat degenerative diseases like aging, Alzheimer's disease, neurodegenerative disorders, cataract, inflammation, etc. So the statins, atorvastatin, forms the base for all this cholesterol related or fat related diseases. In India, almost 95% of the prescriptions which are used in the Ayush system were having more benefits to the mankind. So it is essential to identify and characterize isolation has to be done in the phytochemical or phytoconstituents present in the medicinal plants. So these are the few examples how this taxol derivative and this vincristine discovery, this Taxol derivative, paclitaxel, and this is from the camptothecin, and this vincristine derivatives, how this has influenced and how it has prevented various or extended the life of various cancerous patients. So during the discovery of these compounds, the aim, the scientists who were involved in the research had come across hurdles like adverse effects, in terms of adverse effects. Even then, they could complete this because of its more beneficial effects in terms of killing the cancerous cells. Similarly, when we see the drug discovery of vincristine and vinblastine, again, the discovery has been planned for anti-diabetic. But when they come across the in vitro cytotoxicity studies, they could found that they have extended this. Similarly, this isomer of the derivative of this from this plant, another beautiful drug, vinblastine, also got into market. So why these few examples I have quoted is, even when we are doing some research, we might be missing some molecules, or some interesting findings in our research help us to come out with a new molecule. Am I audible? Excuse me. Am I audible?
Ma'am.
Yes, ma'am.
Thank you. Somebody, some talk I got from Priyanka, I think. Priyanka, you want to say something? Priyanka is there? Shall I continue? Shall I continue?
Yes, ma'am.
So these two discoveries, whatever we are talking previously from the natural products or the plant products, make all the scientists involved in the biology or involved in science. We have to be more dedicated or in our findings. Like if we get some accidental findings also, we need to monitor that. So whenever we are doing a research, we may get in future a beautiful molecule like this. So this is an example where the research has been planned for some other purpose, like, and then how it has ended up with a new moiety. And if you take this morphine derivatives, if you can take this wonderful drug and its derivatives in case of surgeries, the patients will be packed with some dermal packages, and it will be intradermally, it will be given, for example, in case of surgeries involved with women during a hysterectomy. This will help you, this kind of analogs will help them to bear with the, or bear with us, they will not be knowing the pain or surgical pain for at least some 24 to 48 hours. So this is a thing which I have seen practically when a hysterectomy has been done with my close relative, where they have given a dermal patch of this beautiful analog, and the patient age, in a case study, I have seen the patient aged 71, and due to this beautiful drug, that particular woman could able to bear the pain. So like this, we are, as a pharmacist, having responsibility in bringing out new ideas. I'm thanking, like I was with the doctor and all. So that this is one kind of case study I can say where the morphine analogs plays a greater role. So we are not simply going across the drugs and therapy. We need to know about their benefits in the practical or their benefits in day-to-day life. So along with the vincristine and other chemotherapeutic drugs, we are able to treat many of the cancers like lymphomas, breast cancer, lung cancer, etc. So this is what I have talked earlier. The discovery of paclitaxel from the
Ma'am, voice is not coming.
Am I audible? Am I audible now?
Yes, ma'am.
Sorry, ma'am, but your voice is very slow. And
Kindly wait, please.
okay, no issues, ma'am.
Am I audible now?
Yes, ma'am, you are audible now.
Okay, thank you. So coming to the steps involved in the phytochemicals from the plants, what are all the things we need to concentrate? Let us have a look into that so that the students who are planning for the natural products are my fellow friends. Where and all we need to have a concentration is when we are going for an extraction with the plant, we need to know about the nature of the plant material, its origin, degree of processing, moisture content, particle size. So these are the basic things. And the steps involved in the phytochemical research are plant selection, extraction methods, solvent selection, phytochemical analysis, in vitro methods, bioactivity, guided isolation, and in vivo activity. So let us see this. If you are concentrating in one particular isolation, for example, the plant has to be collected in the plant selection has to be done according to the nature of the phytoconstituents. For example, we are planning for a cardiovascular disease. Then we have to concentrate on the family which contains or which has been reported more of that phytoconstituents. So if you take the apocynaceae family, like we got anti-cancer drugs like vincristine and vinblastine. Similarly, during a plant selection, there are two things we need to concentrate: the nature of the study, as well as we have to concentrate in the time duration of work. So when we are going for a duration with the short period for like UG projects, we can take like root extracts. Why root extracts means? In case of root extracts, we can avoid the interference of other colored compounds like chlorophyll. So when we are going for the plant selection, the first criteria what we need to understand is the nature of the work. Based on the nature of the work, and the duration of the work, and whatever the biological activity we are planning, we can select. So initially during the plan selection, we have to concentrate in 2 points. Number 1 is nature of work, and number 2 is duration of work. The extraction methods and solvent selection, phytochemical analysis, in vitro methods, bioactivity guided isolation, and in vivo activity. These are all the things in a phytochemical research. So when we are doing, the plant can be selected based on 4 methods, like, excuse
Ma'am,
me,
slides
whether
are
my
not
screen
moving.
is moving.
No, ma'am, slides are not moving.
Slides are not moving, huh?
Yes, ma'am.
Sorry for the interruption. I'll connect within 2 minutes. Selection, we have to proceed. Plan selection. So we have to take 4 things. One is the traditional usage. This we can get from the traditional chemotaxonomy. So when you take a chemotaxonomical based plant selection, the previous research got having a veterinary research laboratory also holds a big role. When you are isolating a particular constant, for example, we are able to isolate a particular constant like triterpenoids or phenolics in a previous plant species. The same could be tried in another species belonging to the same genera. That means you are working with some Oxymum basilicum, means you can work with other species of Oxymum where you can get the phytoconstant or more beautiful phytoconstant which can avoid the adverse effects. So this is one way we can focus our research. Then based on the phytoconstants which we are quantifying. So when we are quantifying like phenol genus, and we are going for a quantification with well-known method like total flavonoid content, for example, we are able to get a beautiful amount and than the standard, or we are able to meet out with the standard, means that particular phytoconstants or that analysis can be taken as a preliminary step for the further movement, or further we can proceed with that. And this has been already told. We need to concentrate on the solvent based on the extraction method. So when we are going for a maceration, or when we are going for a percolation, we have to concentrate which type of solvents we are using. And when we are going for any pigments or polar constants, we need to take care that the phytoconstants are not getting degraded because of higher temperature. So we need to know about the dielectric constant and their way of separation. So when we are taking, for example, you need to isolate terpenoids, you can go for a chloroform extract. When you want to isolate flavanols, you can go for the polar solvent extract. So these are the various methods like plant tissue homogenization. That is, you can do a powdering with the help of a homogenizer. Then the serial exhaustive extraction methods, where we have this method has to be taken care for the thermolabile compounds where it get degraded. So when we are isolating a thermolabile compounds, we need to go for other cold extractive methods. So this oxalate extraction methods, when we are planning from extract from non-polar to polar layer, we can go for a oxalate extraction where this isolation of will be done if in the solvent extraction we are extracting plant product. The plant product is same. For example, you are taking 500 grams of the root material. You are using a solvent like pet ether. You are extracting, so it is equal to the fractionation. So the pet ether extract removed phytoconstituents can be got. Once again the plant will be dried, the plant will be repacked, and again you will be going with the next solvent like chloroform, ethyl acetate, ethanol or methanol, and then aqueous extract. So this oxalate extraction is easier in isolating the phytoconstituent when we are knowing about the polarity. Maceration, cold maceration extract. So when you are going for cold maceration, when we are having more number of flavonoidal constants. So decoction, these are the infusion and digestion sonication. So these are the various methods of extraction. So when we are doing a sonication, the powder particles will penetrate, the solvent will be penetrated because of the higher energy, and it will be able to, the solvent can penetrate and get the plant constituents in a higher amount. So this is one disadvantage is the active constituent through formation of free radicals, they may go for undesirable changes. So these are the various methods. So far we have discussed about the various methods of extraction. So once your extract is ready, you can go for quantitative analysis, as I have said earlier, like total flavonoid content and total phenolic content by general or by standard methods using FC reagent and will be a eye opener or a start. A scientist can start with this GCMS analysis by knowing about the compounds present. For example, your spectra is having a difference of 14 in its mass number. You can expect like there are more number of CH2 peaks, so that there will be more fragmentation. So the non-polar constituents are more like that. We can compare the GCMS analysis with the already available libraries, and we can know about the nature of the plant which we have selected for the study. So HPLC analysis and HPTLC, when we are doing a thin layer chromatography, we are getting we are getting the bands, or we are getting more number of spots. The same can be adapted for HPTLC, where if a particular constituent is present more, this can be planned for quantitative analysis. For example, you are having a more amount of one particular compound. You can compare with the standard in the HPTLC, and the peak uptime will give you a way for quantitative analysis. That means you can, if there are beautiful phytoconstituents like secondary metabolites like, for example, like lupial and betulin, you can focus on HPTLC studies. So what are the other method of in vitro antioxidant activity? So now, so far we have seen about the extraction by all methods like depending upon the plant material chosen and the duration of work. Already I have discussed. We have discussed with the hard percolation method and the cold maceration method mainly. Coming to the in vitro studies, there are a beautiful, like the in vitro study, you can plan. For example, the DPPH assay, if there is a decoloration, most of the in vitro antioxidant activity are based on the decoloration methods. So once you get more amount of decoloration in your particular extract, that particular extract can be taken for in vivo activity. So when for example, we are doing a in vitro antioxidant activity for a extract, for 4 extracts. There are 4 extracts like pet ether, chloroform, ethyl acetate, and methanol. If your ethanolic extract, alcoholic extract is showing more activity with the selected methods, that particular extract can be screened for in vivo. Then that particular extract can be extended for bioactivity guided fractionation using column chromatography. So these are the advantages of this in vitro. You are focusing on anti-cancer activity means you can focus with the MTT assay and SRB assay, where you can go for short-term studies, cytotoxicity studies, as well as long-term cytotoxicity studies. So this will be a route for continuing your in vivo activity. So that means that the short-term cytotoxicity activity in one day, if it is giving a more potential result with your inhibitory concentration, the same can be extended for long term. If you are getting both short-term and both short-term and long-term a high positive result, the same can be extended for your in vivo methods. So this way, the in vitro activities will help us to proceed or to take to select one particular extract if we are having more number of extracts to go for biological activity. Coming to the isolation. So when we are talking about the earlier slides, I have told you about the isolation of morphine, isolation of various other compounds like paclitaxel, camptothecin, etc., vincristine, etc. So the normal conventional method, which is easy, is column chromatography. The normal phase column chromatography, we will be doing the elution by taking a polar system, and we will be using from non-polar solvent we will be using, and the solvents, the elution will be from non-polar to polar. So I want to talk here for 5 minutes. The column chromatography is a simple technique where and all it can be used in case of research. The column chromatography is helps you when you are knowing that there are 2 particular constants present and their RF values are very closer. You cannot go for any other conventional separation methods. The column chromatography plays a beautiful role. When we are concentrating on one particular extract, the areas where we need to concentrate is the elution part. When you are eluting with the solvents ranging from non-polar to polar, we are starting, for example, we are starting with the solvent like N-hexane or petroleum ether. Next, we are adding some solvent like chloroform. In the fractions, we are taking a column and we are taking the stationary phase here and we are packing this. The column packing can be done by 2 methods. One is wet packing and another one is dry packing. So easiest way is wet packing. And when your mixture is loaded here, this is your mixture. You are taking other solvent mixtures and you are collecting the fractions. When we are collecting the fractions, we have to concentrate on 2 parts. One is the fraction pooling. That is, we have to check this TLC. The fractions collected will be checked in TLC plates. And wherever you are having the same or similar spots, it has to be pooled together. So the tediousness is in the column chromatography is we should not admix any other improper fractions. That is, we are collecting by 25 ml means we have to concentrate in the proper cooling. That is, mixing up of the fractions should be taken care. And coming to the isolation, the fractions, once you get the solid residue, yes. Is there any problem? Shall I continue?
Yes, ma'am. Please continue.
No, any problem is there in audibility, ma? Okay, I'll proceed. So the column, the mixing of the fractions should be taken care. This is what, as a researcher, those who are working with phytoconstituent, the main problem is when you are admixing the improper fractions. And these are the various methods of column chromatography. When we are having a facility like flash column chromatography or vacuum liquid chromatography, our separation can be done with more ease than the normal column chromatography. So the advancements in the normal column chromatography or the gravity column chromatography, flash column chromatography, vacuum liquid chromatography, etc. So coming to the methods of detection. See, when you are viewing doing the separation by UV methods, IR methods, and C13 and H1 NMR methods. Once your phytoconstituents are ready, you are getting a product. That product you are checking by TLC, and you are able to get that as a single phytoconstituent. That we need to subject it for UV studies. If this UV studies help us when your extract is having more number of flavonoids. For example, if you are telling that your phytoconstituent is a flavonoid, then definitely your project or your report should contain a UV graph where there will be two characteristic bands A and B at the UV region. So if your extract, you are subjecting your extract for UV studies and you are getting A band and B band. These two bands indicate that the plant extract which has been taken for the studies contains definitely the flavonoids. Coming to the IR and C13, we are having 2D and we are having depth, NEPT and all in this C13 NMRs, where this gives how many number of CH2 peaks are present in the compound. So how many number of methylene bridge is present can be identified by T13.
जो सारी सारी चीज रहती है जो एकदम मतलब the thing that is very well known.
So C13H1 mass, all this spectral analysis will help you to identify the phytoconstituents isolated. Coming to the new advances, we can, if you are having a crude extract, that particular crude extract can be automatically isolated when we are going for gradient elution using this SEP box. So this apparatus will give you a compound isolation easily. So these are all the hyphenated techniques. So when we are having a preparative HPLC, in case we are knowing that that particular plant is having the secondary metabolite carbohydrates more, means we can choose a preparative HPLC where we can isolate that particular secondary metabolite. So hyphenated HPLC, hyphenated techniques link will help us to avoid the tediousness. Nowadays, there is other technique, flow injection analysis NMR. This instrument helps us to get the analytical report of the component or phytoconstant easily. So coming to the drug discovery, there are 3 stages. One is the pre-drug stage, where the plants are selected, and once the plants are selected, that is the pre-drug stage. How the plants are selected has been already discussed. So the medical information based on the traditional as well as ethnobotanical information, the plants are selected. Coming to the second stage, it is a quasi-drug stage where the plants are extracted and the components are isolated. Once the compound is isolated, and this may lead to a lead molecule, which will help you for further drug development. Then the last stage is the full drug stage, where the natural compound, which was isolated, can help in developing a new compound for the particular study chooser. So when you are spending years from secondary metabolites, we have to cross across the preclinical and clinical trials. So we have to spend some years similar to the synthetic molecules. So these are the advantages in plant drug discovery. So finally, coming to the summary and conclusion, why natural product is required? Because we cannot imagine the molecule. We cannot imagine the combinatorial chemist like nature. So the architecture done by the plant, the phytoconstant, cannot be imagined by the human brains. So we have to take this into consideration. That is the first point. Nature is the best combinatorial chemist, and it provides numerous clinically useful drugs. So various challenges encountered in the plant extraction is more number of phytoconstant present and the tediousness in isolating them and the lower amount of the constant present. 1/10 of the flowering species has been isolated or taken for the study so far. So there are various species in India that has to be studied. Even people are working with more number of plants. There are unexplored plants which we can come out with the beautiful drug moiety. The relevance of natural products in case of innovative drugs has to be studied extensively. Technological advances like earlier discussed, like FIR, NMR, and other hyphenated techniques can help us to overcome these problems. So these are when we utilize all these resources, we can come out with a beautiful drug. So thank you for your patient listening. If you are interested, I can share some of the tips in natural product research. If you are really interested.
Yes, ma'am.
Any
Yes, ma'am.
doubts also?
Sorry, ma'am, can you please repeat?
Yeah, if you are if you are doing any plant research, like if I could help from my side, I'll be more happy. I have some practical tips in like carrying out like process validation or small tips in isolation and all. Any students or any fellow mates are interested means I can help you if I could do it.
Yes, ma'am. Surely, surely. We will.
Can I take another? Yeah, somebody asked about DPPH assay, diphenyl picryl hydroxyl. That is a Mansi has asked. Okay. That is an assay. It is a decoloration assay. You can do it by, if you have an ELISA reader, you can get 12 into 8, 96 readings at a stretch. Mansa, I'm telling to you only. So if you are your college or you are provided with a ELISA reader, we can do the extraction and incubation with the help of a biotechnological department, where the ELISA reader helps us to complete the repetition of 12 readings or 12 readings at a time for a particular extract concentration. So, for example, we are taking DPPH assay. Each in vitro antioxidant assay, we need to take a particular standard. So if you take a DPPH assay, you need to take ascorbic acid as a standard. If you take other ABTS, you have to go for fruitine flavonoid as a standard. So in the DPPH assay, if you are not having a ELISA reader, you have to go with the UV visible spectrophotometric analysis. There also we can do the reading, but we need to repeat it for 6 times and get the average out of it. Hope I have answered your question.
Excuse me, ma'am, I have a question. Can I ask?
Yeah, please.
Ma'am, for the natural product or the plant extraction, if we have to go for perform the GCMS analysis, so many GCMS have the chemical libraries there. But for the plant extraction, GCMS required natural library. So can you suggest the natural libraries for GCMS for plant extraction where is available in India or in Maharashtra?
Yeah, NSIT one library is available, and we have online sources. I have the databases. I will share with you. Like I'll share my email ID. You can share with your doubts can be posted there.
Okay, ma'am. Okay.
Okay. Any other doubts? No doubts?
Ma'am, many students have requested to share the PPT because in the middle of something there was a glitch, na? So students are requesting to share the PPT.
Okay, definitely I'll share to the IQAC coordinator. You can get from them.
So much, ma'am.
I'll share to Abhay sir, no? Correct, no? Or Gajanan sir, I will share my PPT.
Yes, thank you, ma'am.
Okay, I'll send through mail. You can
Any
take
other
it.
participants who have any queries, please type your question in the chat box. Or you can unmute yourself and ask the question.
Okay.
And there is one question. Is there any database available to know which plants are explored and which are not?
There are no, I hope, to my knowledge, there are no such databases. Only thing is, we are having review articles. For example, you are aiming at one particular plant selection. You can go to the Google, generally Google Scholar, and if you type the species or genera, you will get n number of PDFs. In that, if you type review, whatever work has been done so far, it is a normal way. I don't think there are databases for unexplored. Explored may be available. We have lots of past, present, and future concepts, drug discovery from natural products. So that review articles, if we focus, we will get the natural product results and reports are there. In that, we will be able to come to know what are all the isolated compounds so far. And unexplored plants, I think I don't know. Yes, please.
Ma'am, there is one more question.
Yeah.
How can we design ratio for different extraction methodologies?
Can you repeat the question, please?
Ma'am, the question is how can we design ratio for different extraction methodologies?
ratios for different extracts or in column chromatography. Whether the question is related with the isolation part or the straightaway in conventional extraction part.
Prajakta, can you be please precise with your question?
Yeah, when it is column chromatography, the ratios are like optimized, like we are going by trial method. When we are going for extraction, you are taking a powder material of 250 grams, for example, your solvent should be approximately 5 times, that is 1.25 liters. I'll repeat my answer. If you are going for extraction, the powdered plant material, if it weighs 250 grams, the solvent should be 5 times, 1.25 approximately. Based on the nature of the plant material, there will be a slight variation. This is the ratio we are fixing generally for extraction. And in case of isolation, we have to go from non-polar to polar, and it is a trial method, again based on the fractions what we are getting and the TLC results.
For in column chromatography, if we go from non-polar to polar solvents for the isolation of the phytoconstituents, then first of all we have to run all the non-polar solvent through the extracted material, or we have
Hello?
the ratio.
Okay. This is based on the length of the column. Your column weighs 50. For example, your column total capacity is 50 ml. You are taking a N-hexane or you're taking a pet ether, it has to run through the whole column. So 50 ml, if it one fraction you are getting means the column, the solvent has passed through the various layers of the column and you are getting the fractions. And you are starting with 100% N-hexane. You are continuing. You are getting a colorless compound. You are identifying, your lab has a facility of identifying through a UV light. You are able to see a spot means you have to continue with N-hexane for N number of times. So the ratio for increasing the solvent generally is 90 is to 10, 95 is to 5. So you can increase the proportions. Based on the fractions collected, you have to optimize the way of adding the solvent. So this is related with the length of the column.
Okay, thank you.
So the non-polar, if you are running for total 100 ml and you are getting collected and you are viewing, because why I am specific in this means we got a product like this in our research lab. We are adding a solvent and we are able to get the particular constant in triterpenoid. I'm right. In the particular extraction, out of 100 fractions, we are able to separate one compound that was new in that genus selected.
Okay.
Any other questions?
Hello,
Yes.
can I ask question, ma'am?
Yeah.
Ma'am, can you recommend me some books for that?
Books, huh? Okay. Natural Products by Icon. One book is there. Raffle Icon.
Okay, ma'am.
Then
Thank you.
another book is there. You have plenty of books are there. Natural Products Raffle Icon, and you are interested in natural product research, mean you can refer to Newman Craig et al. Alan Butler reviews and natural product libraries. If you are so much interested, you can mail to me so that I can send all the references to you.
Okay, ma'am. Thank you.
Materia Medica is there. Okay, you have Indian Traditional Plants by Kritika Basu, and you have Ayurvedic Medicinal Plants, volume 1 to 5, Indian Ayurvedic Medicinal Plants, Wealth of India. These are all the sources, book sources. Any other questions? Yeah, you can ask. If I know, I will tell you. Otherwise, I will tell I don't know. Otherwise, I will refer and I will tell you. You can ask me.
I think, ma'am, these were the questions. Thank you so much for addressing them.
Okay,
So,
thank
ma'am,
you.
can I take the session further?
I want to, before that, I want to conclude this session by acknowledging you. Shall I take 2 minutes?
Yes, ma'am, please.
Yeah. Once again, I wish to thank Yash Institute of Pharmacy for giving me this wonderful opportunity, which makes me more learning, which makes me anxiety towards learning. I thank all the organizers of IQAC members and all the fellow colleagues and team members, both teaching and non-teaching, and most dear participants for your patience listening. And also, I would like to thank my principal, SNS College of Pharmacy, Satish Kumar sir, who is inspiring us and also encouraging us to do all type of activities. I would like to thank the management and all other teaching and non-teaching faculty who has supported me. I also want to thank my dearest students who is always there to support in all my developments. Once again, I take this opportunity to thank all the participants and the organizers. Thank you one and all.
Thank you so much, ma'am. We are truly, truly grateful to have you once again here with us, and we look forward to further more future sessions with you. Your sessions are almost very enlightening.
Thank you for the compliment, Ma. Thank you.
So let's take the session further. Now I would like to invite Ms. Rutuja Sahaj Rao to propose the vote of thanks.
Estimate guests, respected speaker, Dr. B. Geeta ma'am, distribution participants, and dear colleagues. Good afternoon, everyone. As you conclude this session on the natural product in a drug discovery, it is my privilege to extend a hearty felt vote of thanks on behalf of on the organizing committee on, and all participants. First, I would like to express our profound gratitude to Dr. B. Geeta Madam, Professor at SNS College of Pharmacy and Health Science, Computer, for the outstanding presentation. Dr. B. Geeta Ma'am, your expertise and extensive knowledge in the field of a natural product have provided us with a deeper understanding for how these resources can be abound for the innovative natural drug discovery. Your talk has not only enlightened us on the therapeutic potential of a natural products, but also inspired us to explore the untapped possibilities within the domain. Your ability to convey complex information in a clear and engaging manner has not made this session both interactive and enjoyable. In case the case studies and examples you shared have illustrated the practical applications and the significance of a natural product in a modern pharmaceutical research, making a lasting impact on all of us. I would like to express gratitude to our principal sir, Dr. S.S. Angdi sir, for always encouraging us and providing opportunities to organize such events. Then I would like to thank the esteemed participants for their active engagement and through provoking questions. Your enthusiasm and curiosity have significantly enriched the discussion, making this session creative and dynamic. A special thank to the organizing team at Yash Institute of Pharmacy for their planning and the shameless execution for this event. Your hard work behind this scenes ensures that the session like this runs smoothly and effectively. In conclusion, this session on a natural product in a drug discovery has been a remarkable journey into the fascinating world of a natural pharmaceutics. As we move forward, let us continue to explore an innovative, drawing inspiration from the valuable insights we have gained today. Once again, thank you, Dr. B. Geeta ma'am, for sharing your knowledge and passion with us. And thank you to everyone for your participation and contribution. Thank you, everyone.
Thank you so much. All right.
Ma'am, please accept this token of appreciation on behalf of management and faculty of Yash Institute of Pharmacy.
Yeah, I'm honored to receive the same. Thank you. Thank you so much. I hope we can leave the
All
session
the
now.
participants are requested to join the session at 2 p.m. after lunch.
Thank you. Thank you. And a special thanks to my friend. Thank you.
Thank you, ma'am.